Alright, so today we're going to be talking about DNA history. So we're going to talk about the development of our theories about DNA from our very basic understandings to pretty much the model that we're going to be discussing as we move through. So the first thing we're going to look at is our big questions. We're going to look at what does DNA look like, how does it work, and how was all this determined, really. So where we're going to start is way back in the 20th century, and this is where we knew that traits were inherited thanks to Mendel and a bunch of other scientists at the time, but we didn't really know how they worked, what were they made of. We didn't really have the technology, we didn't have the research to that point. So DNA, as we know today, is a really big part of our understanding of biology, and it's really important for us. It gets at the very basic level of a lot of our life processes. So this was something that we were really looking at. It really started with Friedrich Miescher. He was the one who discovered nucleic acids, which are our bases in DNA. We also started finding out about chromosomes. So we look at cell division, especially during mitosis, and you could see the chromosomes lining up during metaphase and splitting apart in anaphase. And we were able to identify that these things were special and they were getting separated and put into these different daughter cells. So this was something that we knew DNA was made up of. It controlled something, we weren't quite sure what, but it was made up of DNA and proteins. That's something that is very important right here. So it's made up of two ingredients, but we weren't sure which of those two, whether it was the DNA or the protein, was really responsible for inheritance. So then we move forward and we're talking with Griffith, and this is when he started his transformation experiments, and he discovered that bacteria could give certain traits even after they were dead. So there's something going on here that wasn't quite living, but they could still transfer these traits. So he used Streptococcus pneumoniae, strep, and he was using rough and the smooth forms on the right. So the smooth form over here is actually pathogenic, so that's going to harm us. The rough form on the left is harmless, not really going to do much. So what Griffith did was he started out the mouse model, and he would inject living S controls, these living smooth controls, the harmful ones, and what resulted here was that the mouse would die. He then did his control where he took the living rough cells, which were not harmful, and he injected them. The mouse is perfectly healthy. He then took heat-killed S cells, so these cells that would normally be pathogenic, but they were heat-killed, so they have no more of this activity, the proteins are denatured, and that led to a healthy mouse. So there was something in there that was leading to the transformation of these bacteria. And where we saw that is we take the heat-killed S cells, so these ones that were normally not affecting the mice, and we added in these living R cells, something that is not normally pathogenic, but we added in, and somehow, some way, the mouse still died. And we came out with living S cells. So we add in these heat-killed and our living R cells, not harmful and not harmful, and we end up with something that is harmful. So the big question for Griffith was, how do you explain this? So then that was taken on by Avery, McCarty, McLeod. They tried to refine Griffith, and they redid it, and what they did was they exposed just the R strain and the purified S strain protein, and he got the purified S strain DNA, so just the DNA. And only the bacteria was exposed to the DNA, and they found out that the DNA was the thing that was causing this reaction. Okay, so the DNA was the thing that was moving from these heat-killed S cells into these R cells to generate these living S cells. So they found out that DNA was the thing that was causing this. So they took a step further from Griffith's experiment. However, it's not quite enough at this point yet. So we had Hershey and Chase come up, and what they did was this blender experiment, and they used bacteriophages. So bacteriophages are these different cells. They contain protein and DNA, and what they did was they tagged them with radioactivity, whether it was sulfur or phosphorus, and they would tag these phages, and then they would put them in with cells. And what happened is these phages infect the cells, and whether or not that radioactivity was detected in the proteins or in the DNA of the cell determined where this heritable stuff was. Was it in the protein? Was that what was transferring this? Or was it your DNA that's actually doing it? So they were going through They eventually won the Nobel Prize for this. Pretty awesome stuff. And this is just showing bacteriophages injecting their DNA in here. This is how they infect these healthy cells. So again, what they did two separate experiments. First, they had radioactive sulfur, and they added the sulfur, and proteins have a lot of sulfur in them, so that would create a radioactive protein, right? The DNA here is not radioactive. So what would happen is these phages go, they deposit their DNA inside of the bacteria, they would allow these things to mix, and what would happen is the phage DNA goes inside of the cell, and you have the empty protein shell on the outside. So ideally, if the protein is the thing that is transferring this information, you're going to see this radioactivity outside of the cell. If you see the radioactivity on the inside of the cell, then you know that it is the DNA, that is the inheritable stuff. So they went through, they blended this all in a centrifuge, they split it up, and they looked at the liquid that resulted. What they saw was that there was radioactivity in the liquid, which means that it was in the proteins. So what happened here is that the radioactivity that we would expect to see inside of the cell, if it was the protein that's doing the transferring, is still outside. So it's not the protein that's doing this. Now, what they did the second time around was they used phosphorus. Now, phosphorus is a key ingredient in DNA. And so when they put in their bacteriophage with the radioactive DNA, the bacteriophage infect the cell, they transfer the DNA inside, and now what you would be expecting is that the radioactivity that you're looking for is now in the pellet, in that bacterial cell, in everything inside of it, not in the liquid. So when Hershey and Chase did this, they looked at their results from the centrifuge, and they saw that yes, the pellet, the cells, actually had the radioactivity. So it was the DNA and not the protein that is giving this inheritance. So this is kind of a big watershed moment. So we finally figured out that DNA is the thing that is transferring this information. Now, we didn't know a lot about DNA at this point, so we needed to figure out a couple of rules. How is DNA composed? What was its structure? So the first one to do this was Chargaff, and what he did was demonstrate two big rules of DNA composition. One, that all species have different amounts of adenine, thymine, cytosine, and guanine in their DNA, which you can see in here. All these compositions are different. So we could have different DNA codes. We have different genetic codes for each of these organisms. The second thing that he noticed was that in every species, the amount of adenine, right here about 25, equals about the amount of thymine, which is almost 24. Then you have guanine and cytosine being equal, so about 26 and 26. So we have these pairs, and if you guys go through this table, you can see, especially here with wheat, that these are, the sea urchin, that these are very similar between these two things. So they're related in some way. So Chargaff came up with was this relationship between adenine and thymine, and then cytosine and guanine. So this was one of our next steps. And you guys can watch a video your own. The next people to come up were kind of the big dogs in terms of this, were Watson and Crick, and then Francis and Wilkins. Franklin and Wilkins, sorry. So what they were doing were these two competing teams, and they were trying to figure out the structure of DNA. And so they're working very hard to do this, and they're actually collaborating a slight bit, but as they were going through, Watson and Crick were using X-ray diffraction and data that was developed by Franklin, and they eventually came up with this double helix model that you guys hear so much about, came about from looking at X-rays of crystallography of this DNA. And so what they would be looking at right here is this data here. This is what they found. They saw these two curves, and they were looking at this. They're looking at the different structures, everything proposed from Chargaff and back, and they came up with this model, the now infamous double helix model. Now, if you guys saw here that Watson, Crick, and Wilkins all won the Nobel Prize in 1962, you might be wondering what happened to Rosalind Franklin. Well, Rosalind Franklin was, unfortunately, she had cancer before the Nobel Prize was awarded. You can't win a Nobel Prize if you are deceased, at least you couldn't back then. And so she was not awarded. And she's an interesting case. We can talk more about that in class later on. She had a very large part in all of this work. And so we finally kind of figured out, we started to piece together more and more what exactly DNA structure was and how it was involved in heredity. So we started getting our base pairing. We got our adenine thymine bonds, we have our guanine cytosine bonds, we've got our double helix model, and now we're able to actually start getting to work on what DNA does and how does it actually function. So now we're starting to get the structure, the basic structure. And so what they also found later on was that you get these phosphodiester bonds, which is why the phosphate, that radioactive phosphate, works so well in identifying DNA rather than protein. And these bonds interact here, and you get these bondings here. So you have base pairing, where you have A and T, these nucleotide bases, are pairing with one another. A to T, G to C. Those are our base pairing rules. Okay, so you guys can take, again, a look at these videos later on. And what this is showing in this graphic here is how DNA is organized. So we start with our double helix. That gets bound by histones, which wrap around each other and make another kind of helix there, which you get these nucleosomes, which are all these histones bound together. Nucleosome is the whole unit here, all these histones and DNA wrapped around it. Once those nucleosomes and all that start to wrap up together, you get this kind of chromatin fiber, and once this chromatin wraps around and around and around, it eventually becomes what we would call a chromatid. Two chromatids make up a chromosome. So this is what you guys would look at when we talk about genetics, and also this is what we saw in metaphase of mitosis. So what we're going to be moving towards next is how does DNA actually function, and how is it replicated, how does it instruct all the different parts of life. So that's what we'll be looking on in the next lecture.